this stuff you guys have already learned. Some of it will be a little bit of a review. And then some of it's going to be brand new. And so for ultrastructures, we're talking about those things that are attached to the bacteria, usually specifically the cell wall. So with that being said, here we go. So like the other macromolecules, I'm not going to go into this too heavily. There are entire biochem courses, semester courses, just on lipids and membranes. But recognize that we have different types of lipids, complex and simple lipids. They can exist in a couple of different states. So when they're in a single layer, we call these micelles. Big thing about soap. You take fat, you add lye to it, it creates these single layered micelles. Really great for removing oil and dirt off of your skin. That's why you have those things in your detergents like sodium diallyl sulfate, SDS, and some of these other things in your shampoo and whatnot. And then, of course, we have complex lipids. And these usually make up what we call a lipid bilayer. So basically, two lipids together. We're going to talk about single lipids very quickly, or single layer lipids very quickly, and basically simple ones as well. And then we'll go into these different types as well. So for our simple lipids, we see these in different places, not really in the membranes of our organelles and our cells, but say for instance in our serum and in our blood. Example of these would be triglycerides. And so most of the time when we talk about lipids, we think of them as having some sort of fatty acid chain, which is usually nonpolar, so that is hydrophobic. And then some sort of glycerol or other type of complex head to the fatty acids. And these are usually polar, and they can be made of different things. For instance, triglycerides aren't quite as, soluble as some of the others, say complex lipids, but we see them everywhere, and so I'll give you an example of what a triglyceride looks like. You're going to love these. There are 14 of these, by the way. So what we have are basically these fatty acid chains. And then in this case, for a triglyceride, it's just basically a glycerol moiety on the ends. These ones aren't very soluble. These are pretty nonpolar. For instance, if anyone's had your blood analyzed, you go out and eat a big old Kentucky Fried Chicken dinner the night before, a big old hamburger, and they spin your blood down, and you see that layer of triglycerides, that fat in your blood. It's basically these triglycerides that you're seeing. So these are very simple. They can be utilized as an energy source, many different things. So when we're talking about these fatty acids, recognize that this is kind of the Lewis shorthand, so it doesn't get all crazy complex when I draw it out. But at each one of these junctions, here, 14 of these, I'm just going to kind of blow it up on this side so we can see it. At each one of these junctions, there's actually a carbon. So I could put a carbon here, a carbon here, a carbon here. so on and so forth. And with each one of those carbons, when we look at it in this shorthand Lewis form, we know that there are 2 hydrogens attached to each carbon, such that the octet rule is fulfilled. And as you can see, it starts to get really kind of hard to look at after a while. So we assume that at each one of these junctions, there's a carbon with its representative 2 hydrogens all the way through. So these are single carbon bonds. With your triglycerides, we would call this a saturated fatty acid. So anytime we hear about these saturated fats, this is what they're talking about, the difference between saturated and unsaturated. What the hell are they talking about? They're talking about that it's saturated with hydrogens, so that at every carbon you have 2 hydrogens. So on and so forth, within that backbone chain. So these are all single bonds, carbon to carbon, single bonds, throughout that entire fatty acid chain. This is different from unsaturated. The difference is we add in 1 or more double bonds. So, an unsaturated would basically have a double bond between one of the sets of carbons. So in this case, this one would have a hydrogen, this one would have a hydrogen, same with this one, hydrogen, hydrogen. But because this is a carbon-carbon double bond hanging out there, it's only going to have 1 hydrogen off of each carbon. So we take those hydrogens, and those electrons go into forming these double bonds. So this would be unsaturated. 1 or more double bonds. So saturated fatty acids are more congealed. They have a higher melting temperature. Think of shortening. One of my favorite things are lard. Unsaturated have a lower melting temperature. Start thinking of canola oil, all that stuff. You know what canola oil really is? No. It comes from the rapeseed plant. But because rapeseed doesn't really sell well in the supermarket, they changed it to canola oil. Pretty funny, huh? So this would be a nice, simple, single layer type of lipid. Now when we deal with biological membranes or biological lipids, these are usually made out of complex lipids, and most often, but not always, we call these phospholipids. So rather than having a glycerol, it's going to have a phosphate head on it. And so I'll give you an example of this. Here's a phospholipid, if you will. And in this case, they only have 2 fatty acid chains. So for your phospholipid, that is an oxygen, you have your normal fatty acid chains. And in this case, we have this large phosphate polar head. So this one is very polar, meaning it is hydrophilic. It likes water. So one side of this lipid has this big hydrophilic side. The other side, of course, as we know, hydrophobic. So when I go about drawing lipid membranes, rather than draw out these huge, complex Lewis figures, being a white male in science, we represent things in kind of cartoonish stick figures. So, for a phospholipid, I represent it with this big phosphate head and the fatty acid chains. So it kind of looks like this little alien. And it basically represents that structure. So when we're dealing with biological lipid membranes, these are bilayer membranes. So if we think back to how I was representing the various membranes in our gram stain, we see that the fatty acid chains turn towards one another because they are hydrophobic. The polar heads turn out. So when we're dealing, for instance, like with a cell wall of a bacteria, this is going to be the outer environment. Of course, it needs to be polar, so it can interact with the aqueous environment. This is going to be the inside of the cell, or the cytosol with all the goodies. It also has to be polar because this is an aqueous solution. But we get this hydrophobic space in between. And we have to think about that. So if I'm a bacteria and I'm producing a molecule, say a great big carbohydrate chain, and I'm trying to excrete it to the outside, I have to have enough energy to push it through this hydrophobic space so that we can make it to the outside. So we always have these polar sides and then this hydrophobic side. Now with single layer micelles, they can be made of both complex and simple, it doesn't matter. What we see rather than with this bilayer, they form these circular micelles. All their fatty acid chains turned in. So these things are really lipid soluble. They can move through membranes, they can absorb oil. So if I'm basically washing with water and soap, this soap is made of these micelles, all the oil and dirt get trapped inside the micelle, helps remove it from the surface of the skin. Membranes, great things. Back in the old days, we thought they were just rigid, super static, didn't really do much. But now we know that these lipid membranes which we see on the outside of cells, so composed of cell walls, on the outsides of organelles, things like your mitochondria, all of that, nucleus, Golgi apparatus, rough ER, all made of these bilayer lipid membranes. And so when we talk about these lipid membranes, there's kind of an old school way of describing it, and I like it, so I stick with it. And we call this the fluid mosaic model of membranes. So what does that mean? Well, that means they are ordered, they have order, and this is due to that lipid biochemistry. They're always going to have this polar side and this hydrophobic side. So they always order themselves, okay, whether in a micelle or a lipid membrane. But they're not static. So they're fluid in that things move through them. So metabolites, carbohydrates, proteins. So we have this really nice structure, but at any time things can move through it. So for instance, we can have various membrane proteins that are either, that are either anchored, what we call anchored into the membrane, or they might actually go completely through the membrane. And so this would be a trans-membrane protein. And then we can have all sorts of other things inserted into them as well. So the things that we see, for instance, are proteins, lipopolysaccharides. We have big mechanisms like transporters and channels. And so transport and being able to move stuff through this membrane is a big thing. So, as far as the rigidity, organisms need to have their membranes stay sort of, well, structurally solid to do what they need to do. So what we find with membranes is they have a few different properties, of course. So first and foremost, they're a permeability barrier. Keeping the bad things out and the goodies in. They can have all sorts of transport, both passive and active transport. When I go through membranes in biochemistry, we spend about a week just dealing with all different types of transports. They can be really complex, really fun to play with. And then one of the big ones we find is they generate this proton motive force. And that'll kind of help us lead into metabolism a bit, everyone's best and favorite subject, I know. So how do we keep these membranes rigid? Well, it all depends if you're a eukaryote or a prokaryote. You'll kind of recognize some of these terms, of course. So at times they need to have a very rigid membrane, and at times they need to have a very fluid membrane. So they can, at times, mess with this and tweak this so that they can have a more rigid or less rigid membrane. If we increase the fatty acid chain length, we end up increasing rigidity. So that's one of the ways we see it. Then we can also increase another type of membrane molecule, basically called cholesterol. Increase cholesterol, increase rigidity. If you're a bacteria, it's a little bit different. Same sort of process about membrane rigidity. Same thing, increase fatty acid length. And you increase rigidity. But they don't have this thing called cholesterol. That's a much more complex molecule than what they have. But what they do is they increase this thing called a hopanoid. And same thing, it is a primitive type of cholesterol molecule. And that also increases rigidity. So at any given time, they can add cholesterol or hopanoids, or they can increase their fatty acid chain length or decrease it, depending on what they do. And as far as these membranes go, really the only thing that diffuses through it is water. Otherwise, pretty much everything else has to have a specific transport or channel or pump to make it go through back and forth. We have these things called aquapores in our membranes that allows the water to kind of make sure we have the correct osmolality so that we don't end up blowing or shrinking the cell up. So let's talk about this proton motive force. let's say I just have some sort of organelle. This could be similar to a mitochondria. It could be a prokaryote membrane because they don't have mitochondria. But here we have our membrane. So I'm just kind of doing this in a very general sense, then we'll come back and talk about it in a much more specific sense. And this membrane has this potential to be electronically charged. So we have specific pumps within the membrane that take hydrogen, remove an electron to create these proton pumps. And what we can do is we can pump these positively charged hydrogens or protons to the outside of the cell. The free electrons that are freed up remain on the inside of the cell. And so we create, if you will, this electromagnetic imbalance. It wants to equilibrate. Charges attract, right? So these positive charges want to come back through due to this electromagnetic attraction. So when we talk about a proton motive force, usually it's during energy or metabolism. So for instance, we can now use this proton motive force because these protons really want to drive through the membrane. We actually have these things called ATPases, and they use this proton motive force to drive the synthesis of ADP into energy forming ATP. These are those nucleotides that are also energy forces. So by creating these electronic imbalances, we can try to let that equilibrate. And it's actually an energy source or a driving force that we can use to produce energy. So when we talk about that, that's more or less what we're doing. So as far as some of the transports we see, I'm just going to describe a couple of the more or less passive transports. I'm not going to go into all of them. So when we're talking about passive, recognize that no energy is needed in order for this process to take place. It's usually driven in a couple of different ways, based upon ion concentration is one way, but we can also do this based upon thermodynamics. Everyone loves chemistry and thermodynamics. We couple one reaction that's less thermodynamically favorable with another reaction that's more thermodynamically favorable, and it kind of drives us in a passive way. Then, of course, we have active transport, and it's just the opposite. Needs energy, usually in the form of ATP. I'm just going to describe some of the passive ones for now, and then if we need to, we can come back and be more specific as we jump into them throughout the semester. And I'm sure you've heard these before, I'm hoping. And so I'm going to give a schematic here, general schematic, which I'll use for each description. Here I have my transporter system, whatever it may be. So here's my membrane. Here's my transporter. For a uniporter, we basically move one substance in one direction, either in or out. So for instance, I'm just going to make this a big square molecule. A uniporter basically just moves one substance in one direction. Then we have the antiporter. Two substances, opposite directions. So if I rewrite this, I would now have, for an antiporter, two different molecules going in two different directions. And then the last type of transporter we can see that is very common throughout prokaryote microbes and eukaryote microbes are the symporters. Two substances, same direction. So I'm just going to reverse those arrows as such. Now, active active ones have all sorts of interconnected molecules at the base of this membrane that are there to help utilize ATP and generate the energy needed. Some of these have all sorts of gates to them, so we don't want things to flow through, so we can gate them. One good way to do that is kind of like the ball and chain. Think of your toilet plunger. So I can basically, as I need to, pull the chain up and the ball clogs the transporter. I can let the chain go and the ball drops and stuff starts to flow through. So we have all sorts of variations on this practice. So with that being said, let's describe some of these structures and ultrastructures and see how far we get. When we start to talk about growth and generation, we'll come back and talk about how these organisms get their morphology. But when we talk about their morphology, we've gone through this already. You can either be cocci or coccus, singular and plural. Of course, these are spherical or round. We can have bacilli or bacillus for singular. Those are rod shapes. You can have spirillum and spirochetes. And we think of these as spiral. This one is a larger spiral, usually with a center molecule. Spirochetes are a little bit smaller. Spiral organism, that means the spiral is closer together. We can have things like appendaged organisms. They kind of look like a gourd with a handle hanging off of them. And with appendage, they kind of have like a tube or stalk hanging off of them. And then we can have filamentous. And this can also be called streptobacilli. That's basically rods in chains. And they start to look so long that they look like filamentous hairs when you look at them. Sometimes like the Bacillus megaterium, we stain and the spore stain has that filamentous look. So these are the normal shapes. Now one thing I'm going to kind of go off really quickly and describe, and you may have kind of already noticed it. When we have a small cap bacillus, this is an adjective that means rod shaped. When you see the large cap bacillus, this is a genus name, like Bacillus anthracus or Bacillus megaterium. So the scientists found this group of bugs, they all were rod-shaped, and they went, oh, we'll just name the genus Bacillus because they're all rod-shaped. So it can be kind of confusing at times. So if you see it with the big cap, you know it's the genus and that they're all probably rod-shaped due to their name. The most famous one, of course, is Bacillus anthracus, causes anthrax, one of my favorite organisms out there. So with that being said, we have cell arrangement as well, and we've talked about this in lab already. So it's not the shape of the cell, but what we see as far as arrangement. These are always prefixes. So we can have strepto. So for instance, streptococcus. This means that we have chains of organisms. Streptobacilli, streptococcus. The Greek term staphylo means grape-like or grape-like clusters. So like Staphylococcus aureus or Staphylococcus epidermidis is a great example of that. And then they can hang out in 2s and 4s. As little diplos or as tetras. Usually these are cocci that do that, but I'm sure there are some rods that do that as well. And of course, we've seen that wonderful gram-positive, gram-negative cell membrane, and I'm just going to reiterate it very quickly so we can see the differences between the two again. So I'm sure you're going to see this ad nauseum somewhere on the test. So for our gram-positives, the key thing is they only have one membrane. This would be the cytosol, or the inside. This would be the environment. And they have these wonderful layers of peptidoglycan. This is kind of a protein sugar-like moiety. And then this is held together by a specific type of acid called teichoic, that is an I acid. Our gram-negatives, of course, have 2 cell membranes and very little peptidoglycan. And in this case, our peptidoglycan is sandwiched in between. This is going to be the environment on this side. And it's going to have all sorts of proteins anchored in the membrane and everything. On this side is the cytosol, or the inside of the cell. Now this space that is sandwiched in between here is called the periplasmic space. And so when we look at these, you can see there are these distinct differences between the two. 2 membranes. Why is that evolutionarily important? Because they're super tenacious, super tough. Think of those gram-negative bugs inside your gut. They have to be excreted out into the environment, hang out in the environment for a while until you touch it and eat it. And then it's got to go through your stomach acids into your intestines and still be alive. So it's really nice to have this double membrane. So we have this environmental protection going on there. But it also makes it really tough as far as antibodies and antibiotics and being phagocytized by white blood cells. So it kind of protects it against all of this. Those were those really critical gram-negatives they were talking about in the video. They have this extra kind of tough coating to them. So why is this advantageous for the gram-positive? For our gram-positives, think of it as being sticky. So it can adhere to your cells. Think of those streptococci that hang out in your lungs, Streptococcus pneumoniae. You're coughing and sneezing, and all this phlegm is always moving around. You have cilia on your respiratory cells trying to get rid of it. And so they're just trying to hang on. Gram-negatives, really tough and tenacious because of the dual membrane. Gram-positives, really sticky because of all that peptidoglycan. So think of it as an evolutionary advantage one way or the other for these guys. So associated with these cell walls are a bunch of what we call ultrastructures or superstructures as well. I'm going to list them, and then we're going to go through them one by one. We have flagella. Flagella, of course, we'll talk about, are used for movement. We have these things called fimbriae. Fimbriae plural will have an E on it, so I call them fimbriae as a plural thing. It'd just be frimbra if it was a singular. We have these things called pili. All three of these are very related. Related in the structure, related how they're anchored in the membrane, but they don't have all the same sort of function, unfortunately. Then we have these things called S layers, which is a specific type of protein that kind of acts like Kevlar on the outside of cells. One of my favorites, capsules, and this thing called biofilm. Together, they make a really wonderful term, glycocalyx. Glycocalyx, say that 5 times fast. And then we have things like storage units. Sometimes these are called vacuoles or inclusion bodies. And they're usually for storing energy. So let's start with our flagella first, and then we'll work our way through. First, we have to recognize that there is actual motility in prokaryotes. This is true motility, and it's different from what we call Brownian movement. As you've probably noticed when we stain and look at microbes, we don't put a cover slip on them. We don't keep them in solution. And the reason why is because of this Brownian movement. These bugs are so small that H2O basically moves them around, makes them look like they're wiggling. But all it is just the water molecules bombarding the bacteria, so they kind of shake. So this is not a true movement. Okay, this is a false sort of motility. When we're talking about real motility, usually it's associated with the flagella. I think of this kind of like a protein whip on the outside that they can whip around, and it allows them to basically move based upon this. So this flagella is a protein strand. So it's made of protein, and it's anchored into the membrane. So it's completely associated with the membrane. And we can describe them based upon the locations of the flagella. So we can describe the first one as being monotrichous or polar. So if we just kind of have an E. coli here, here's my bacteria. We're basically going to have one flagella coming off of one side of that organism. Then we can have bipolar, really depressed E. coli that swings both ways. Sometimes it's really happy, sometimes it's really sad. So basically a single one on either side. Then we can have what's called lophotrichous. Think of it as many on one side. Lopho meaning kind of location. And then we have peritrichous. Anytime you hear that term peri, that prefix, think of it as being everywhere. And so that's what we see. So lopho on one side, bipolar, one on each side, or excuse me, monotrichous. Lopho meaning multiples on one side. Peri just has them all over the place. Now there are other types of motility that we talk about here and there that aren't associated with flagella, but we don't really go into them as much. One of those would be contraction. one of those would be gliding on a sugar that we'll talk about, basically that glycosylates. So now the thin braid is a little bit different. So it's a protein, structurally similar to a flagella, but it is not used for movement. It is anchored into the membrane, like the flagella is anchored, so it's basically an anchored protein as well. But rather, it's used to basically adhere or attach. Or adherence. And the last one that's very similar to this are pili. Once again, not for movement. But for adherence or attachment, but a special type of attachment through this type of reproduction called conjugation. And so for conjugation, what we see is that these cells connect with a pili, or pilus in this case, and genetic material can go from one organism over to the other. So it's this way of kind of blending the cells together so that we can move DNA from one organism to the other. We see this specifically in gram positives. We don't see them really in gram negatives. And the last subject we're going to talk about for the night, before we run into our cool food lab, are capsules and biofilms. So these are sugar carbohydrates, if you will, and they are associated or attached, if you will, to the cell membrane. And they just make this really kind of sugar mucoidy substance around it. So these polysaccharides, if you will, are used to be sticky and attachment. They're also used for protection. So it's like putting a Kevlar sheet, if you will, sticky Kevlar sheet around the outside of the cell. And so it helps protect from antibiotics. Once again, antibodies, which I'm just going to shorthand ABS, protects from being phagocytized by white blood cells. And this, as well as some of these others, we consider a virulence factor. So flagella, motility, and some of these other things we also consider a virulence factor. Virulence, the act of being infectious or pathogenic. So if you're a virulence factor, that means it increases your infectious nature, makes you more infectious. So a good example of this, Streptococci pneumoniae. So this guy produces a lot of capsule. And it's the #1 causative agent for double lobar pneumonia, usually with effusion, nice liquid that builds up outside of your lungs. If it has the capsule, let's call this capsule plus, it's virulent and infectious. But I can basically go in and knock the genes out for that capsule and make the capsule go away. So we can make it capsule negative. And it becomes non-pathogenic. You can drink it by the gallons. Doesn't matter. But by adding that sugar capsule, it now becomes much more pathogenic, much more virulent. And along the line of this, basically very similar and related to it, is the biofilm, which we call an exopolysaccharide. So this just means it's excreted outside of the cell. It has no membrane association. And sometimes it's made in conjunction with the capsule, so they can actually have the capsule plus the biofilm. And once again, this is all for protection. And for community living. It's kind of like a commune. So we used to think that bacteria were sessile planktonic organisms, just single cells that didn't really interact with one another, kind of just hung out in solution. But we know now that they live kind of in conjunction with one another. So when we see these biofilms and the capsule together, that's that glycocalyx. And sometimes if we take a look at these biofilms, they'll have different organisms living within the biofilm with different duties. You said duty. Been around my kids a lot. So here we have this biofilm community. And you can have various organisms located with various jobs throughout this biofilm community. I'll give you a great example, just to end the lecture. I used to do a lot of consulting, microbial consulting as a job, kind of as a side job. And one day I get a call from the Teton County School